Every cell in the body contains the entire genome. However, not all genes are expressed in every cell all of the time. How do cells control which genes are expressed? To understand this, we need to explain how transcription is controlled, explain how translation is controlled, describe how proteins can be modified after translation. Many factors affect the transcription of DNA, including the structure of DNA itself. In eukaryotic cells, DNA is wrapped around proteins called histones so that it can be packed into the nucleus. Some parts of DNA will be more tightly packed than others. The proteins involved in transcription can reach the genes in loosely packed parts of DNA, but not the genes in the more tightly packed DNA. How tightly the DNA is bound to the histones is affected by acetylation and methylation. When acetyl groups are added to histones, they become less positively charged, and so are less attracted to the negatively charged DNA, allowing genes to be transcribed. When methyl groups are added to cytosine bases in DNA, the DNA becomes more tightly packed, and genes cannot be transcribed. Transcription factors are proteins that switch on transcription. They move from the cytoplasm into the nucleus and bind to a specific base sequence in the DNA, switching on transcription of that region. Some transcription factors are regulated by hormones, meaning that until the transcription factor has been activated by the hormone, it cannot bind to DNA. Hormones like estrogen are lipid-soluble and so easily pass through the cell membrane. Once inside the cell, estrogen binds to a receptor site on a transcription factor, changing the shape of the molecule and activating the DNA binding site. Now, when this transcription factor enters the nucleus, it can bind to a specific site of DNA and initiate transcription. The process of transcription produces an RNA molecule from the gene to be expressed. This RNA is called pre-mRNA. Before it can bind to a ribosome and undergo translation, the pre-mRNA is modified. A cap and tail of adenine molecules are added to the pre-mRNA to stabilize it. The pre-mRNA may also be edited by addition, substitution, or deletion. This has the same effect as point mutation and can produce different proteins from the identical strands of pre-mRNA. Translation is the process of synthesizing the proteins coded for by mRNA, but not all mRNA that is produced is translated. It may be broken down in the cytoplasm, for example by small interfering RNA. Small sections of double-stranded RNA are produced in the cell. They split into single strands and bind to specific enzymes. The single strand of small interfering RNA will attract complementary sections of mRNA. Once bound to the small interfering RNA, the mRNA will be broken down by the enzyme. Following translation, not all proteins are active immediately. A protein may need to be activated by phosphorylation. Enzymes called protein kinases regulate protein phosphorylation and so can regulate cell activity. Proteins can also be modified after translation by folding, shortening, or addition of non-protein groups, all of which can be used to regulate how much of an active protein is produced. There are many different ways that a cell can control gene expression, including controlling transcription and translation.